Dr. Landowski is Research Assistant Professor at the Department of Medicine at Arizona Cancer Center at the University of Arizona. And she is a PhD graduate from Montana State University and did postdoc training at U of A and the H. Lee Moffitt Cancer Center at the University of South Florida. And she's going to tell you more about the work she's doing. And again, I've just had the pleasure of meeting Dr. Landowski, I think it was just a couple of months ago. I'm called tenacious, some call me other things, but when I'm after getting the very best for us, there is no stopping me. And thank God there are people like the ones you're hearing today who give up a Saturday and come out here. So Dr. Landowski, I'm going to let you go on with your presentation and your research. Thank you.
So Barbara, thank you for the invitation. It's always a great opportunity for us to get out of the lab and educate the public on what we do. It's also a little bit intimidating for me to be following John Carpton and Dr. McHale from Mayo. I'm the little guy in between. So John is collecting all the data and analyzing huge data sets and producing a tremendous amount of information, which is really fueling a lot of the work that I do, which then goes on to be translated into the therapies that actually come down to the patients. And so what I'd like to do today is to show you some of the paradigm shifts that I hope that I've contributed to, at least in some small way, and some small examples of what the little guys do in between John and Dr. Joe. And you need to know that there are a lot of me out there, because there's no way that I can keep up with these guys. So, first of all, let's see. Yeah. To be or not to be. Is a murder-suicide? It takes a village, and location, location, location. And a lot of these are concepts that both of the previous speakers have touched on. And I'll show you an example of how we've learned this. So, to be or not to be. For many years, everyone has assumed that cancer is a disease of growth. The tumor is growing too fast. The tissue has converted and it's growing out of control. But one thing that we've learned over the last probably 10 years is that it's not always because it's growing too fast. And I think multiple myeloma is probably the primary example of that. It's a matter of the cells not dying when they're supposed to. In your immune system, the cells are recruited, they do their job, and then they're supposed to die and go away. But for some reason, in the myeloma cells, they don't get that message. They don't die and they don't go away. And so several years ago, we examined one of the cell surface receptors that's used by plasma cells and other immune cells as this off switch that tells it to go away, and it's called FAS. And this is, I'm sure that John's laughing at this. This is old technology where we're using a gel shift to identify mutations. And so as you can see, I'm sure I'm going to hit the wrong button here. So these are bone marrow samples, and we obtained bone marrow samples from 54 patients and amplified their DNA. Actually, this is cDNA, amplified the RNA, and looked for mutations in those genes in the FAS coding region. And you'll see these guys where there are two bands, there's two chromosomes, those are normal. But the ones with mutations, you get this extra third band that shows up above, and these are just the controls, the normal and the mutated control. And what we found was that of the 54 patients that we analyzed, 10% had mutations in the coding region and actually in the functional domain of the FAS molecule that would cause it to not work. It would not be functional. So when the immune system says, you're done, die, go away, the cell doesn't get that message. It survives and goes on to produce a tumor. An additional 10% didn't even express the receptor. So there's some additional genetic lesion, which would be identified by the fish that you've heard about, or the SNP analysis that John mentioned. So this is one of the things that really shifted our thinking, that it's not always a disease of excessive proliferation. It's a failure to die. So to be, choose not to be. Another thing that we've learned over the past several years is that when we treat the cells with therapeutic drugs, as Dr. McHale mentioned, it used to be a blast of machine gun fire, and we hoped that the normal cells were less susceptible to the machine gun fire than the tumor cells. Essentially, we treated tumors with poisons, and many of them were derived from natural products, such as doxorubicin from the foxglove plant. And what we found was that we could make more effective therapies if we used the machinery of the cell itself. So in the immune system, when it shuts off the switch and tells it, die, go away, you've done your job, there's a system of a programmed cell death machinery that's activated, and a whole series of events that must occur so that the cells will die and go away quietly. And in the work that I'm showing you here, we found that sometimes the machinery is not all functional, or there are survival genes that are overexpressed. And so these are, again, these are samples derived from patient bone marrows, which we stained the myeloma cells, the kappa light chains, with red. Oops, see, I knew I was going to hit that wrong button. The light chains are stained with red, and the apoptotic or dying cells are stained in green. And so you can see in this patient sample, there's 3 big, fat, healthy myeloma cells. They have no green. When we treat with one drug, with the anti-FAS antibody, we get a lot of dying cells, and you can see the ones that are green.